Thank you all for signing in today for our live Dockside chat. My name is Catherine McCourt. I'm the Communications Coordinator at the Centre for Coastal Studies. And today's topic, in case you didn't guess from our opening song, is All the Small Things, presented by Dr. Agnes Mittermayr. A lot of you know Agnes already. I see some of her volunteers are on the webinar today. But for those of you who don't, Agnes is a marine archaeologist. Agnes is a marine ecologist. Her research focuses on how physical, chemical, and biological processes and interactions shape the available benthic habitat and species composition of coastal waters. Recent and ongoing projects include benthic habitat mapping, restoration of salt marsh and eelgrass habitats, stable isotope studies of food web interactions and trophic niches, which Agnes will explain, and tracing and quantifying the effects of human activities on the coastal community structure. Agnes has a PhD in biological oceanography from GeoMar in Kiel, Germany, and an MS in oceanography from the University of Southampton in the UK. She's a scientist for the Geology department at the Centre for Coastal Studies and a benthic ecologist for the Cape Processes and Ecosystems Laboratory, that's the Cape Lab, which is a joint research effort between the School for the Environment in the University of Massachusetts, Boston, and CCS in Provincetown. So just before I hand you over, I want to remind you all that we will have a Q&A session at the end of this. So please put your questions in the Q&A box, which should be at the bottom of your screen if you're on the PC, or I think it's top right if you're on the smartphone. So we will try those throughout the conversation. If not, we can follow you offline as well. So do feel free to drop us an email. So with that, I will hand over to Agnes for all the small things. Thanks, Agnes.
Hi, everybody. Thanks for tuning in. I'm going to tell you about all the small things around the Cape today. We've all heard it at one point. The sixth extinction is upon us. Books have been written about it. We're losing species left and right. It is predicted that we could use half of all species by the year 2100. We are ruining the planet. This is horrible, absolutely devastating, especially in a time when conservation laws are being rolled back. But not all is lost. We just have to get a little bit more creative about how we want to protect nature and save species from extinction. And one way to do that is to look at how the species relate to each other and their environment. In the Department of Geology at CCS, we're trying to figure out just that. This is a map of East Harbor and Truro. I hope you can see my cursor. So here we have East Harbor. Down here, that's Cape Cod Bay. Provincetown is to the left here. And this body of water has been through a lot over the past centuries. At one point, it was open to Cape Cod Bay, indicated by that black arrow over here. And then train tracks were laid approximately where Route 6A is right now. So it was cut off from water exchange. It turned into a mosquito-infested freshwater lake and later was reconnected to the bay and is now a brackish body of water with a tidal range of a few inches. So we set out to explore the connections between the species assemblages and their environment, like what type of bottom it is. Is it sandy? Is it muddy? What do the currents do? What's the oxygen levels like? Temperatures. So basically all the parameters that have the potential to influence species presence or absence. And together with an army of volunteers, without whom this and all my other projects would not be possible, we picked through gallons and gallons of sand, looking for small invertebrate creatures. Invertebrates are animals that don't have a spine, like insects, squids, spiders, and those tiny little hydrobia snails that you can see in this picture here. Together with this information, we used grain size data, water quality data, bathymetry data of the area that we looked at, so East Harbor, and we developed habitat maps, biotope maps, like this one right here. and we used the Coastal and Marine Ecological Classification Standard, or CMEX for short, to make them presentable. We learned to understand the connections and the relationships between the animals and their habitats. Is there fine sand and not a lot of movement? Then you might find the amethyst gem clam right there. Is it muddy? That's definitely worm territory. Overall, we could explain more than 80% of species distribution just based on the environment. This resulted in a publication, and at that time was the highest percentage of species distribution explained in North America. So we have a pretty good understanding of the relationships between species and their surroundings. We know what kind of environment they need to thrive. But what about the relationships between the species? One way to look at that is to figure out the predator-prey relationships. I'm sure you've heard it all before, food chains, where the zebra eats the grass and the lion eats the zebra, plant eater, meat eater, eat or be eaten, the circle of life. But what if I told you that there is no circle, much less a chain? It's really a convoluted web of strong relationships, chance encounters, missed connections, and wallflowers watching from the sidelines. In fact, predator-prey relationships are so complicated, there's an entire branch of science devoted to it called trophic ecology. And it gets even more complicated underwater, where we can't hide out and observe what the animals are eating or use game cams to keep track of their meal plans. In fact, this food web right here is fairly simple. It focuses mostly on fish. It doesn't take into account the hundreds of species of worms and shellfish we find on a daily basis around Cape Cod. So where to start? Thanks to scientists who came before me, I can now stand on the shoulders of giants and use nuclear physics to untangle food webs and find out how animals are connected to each other. We can trace food webs by analyzing stable isotopes. As Carl Sagan so eloquently put it, we're all made of star stuff. And he was right, because nature is really, really good at recycling. Stardust is made up of by atoms, just like every single thing we encounter in our daily lives. And those atoms, the ones that built you and me, were formed in the Big Bang and are constantly being recycled. Not only are we made of star stuff, we are also what we eat. The food we ingest helps build us, literally. Those building blocks can be traced back to their origin. So what are those building blocks? This is a schematic of an atom, which is what we're made of. This atom is nitrogen. It has 7 protons, the red dots in here, and 7 neutrons, the green ones, in its nucleus. 7 plus 7 is 14. So this is called nitrogen 14 or 14N. It's in the air we breathe. It's in the plants we eat. It's in our DNA. It's everywhere. Now, the Big Bang not only created regular atoms, it also created isotopes. There's the unstable ones, the radioactive ones, and the stable ones, which is what we use to trace food webs. So the stable atoms, the stable isotopes of N14 are called N15. That's because of the additional neutron in its nucleus that you can see right here. So 7 plus 8 is 15. That's where the name comes from, nitrogen 15 or 15N. The additional neutron doesn't really cause the atom to behave any differently, but it does mean it's a little bit heavier, and that's where the food webs come in. This is a picture of me eating my first ever In-N-Out burger. It was an experience. And with every bite I ingested the regular atoms and the stable isotopes. So now, as we all know, what goes in must come out. And the body extracts, of course, everything that's useful from the food we eat. So in total, we are about 65% carbon 65% oxygen, 12% carbon, 4% hydrogen, and the rest is nitrogen and a bunch of other elements. But this is not where it ends though. There are still the heavy and the light isotopes, the regular ones and the ones with the additional neutrons. So the heavy isotopes are a little bit more reluctant to move. It takes more energy to get them going. Like with a big rock, it needs more energy to be moved than a tiny little pebble. So the heavier isotopes, they stay behind, while the light isotopes get flushed out, which means that we are enriched in heavy isotopes. And the higher up in the food chain we go, the more enriched the organism becomes. After all these years, it's still baffling to me that I am built from the ocean. I am the fish fry and the seaweed salad, down to the nucleus of my cells. It's a fascinating subject, but I'm not going to get into any more technicalities here. You're welcome to ask, but be prepared for a very excited scientist to rave about mass spectrometers and electron sources. I mean, we can literally weigh atoms. It's mind-boggling. Anyway, back to the relationships between species. Once we know of the ratio of heavy versus light isotopes in tissues such as scales, bones, and muscles that we analyze, we can start building food web models. A lot of statistics go into those things, and at the end, we hopefully end up with something like that, what you see here. You can see that there's connections everywhere. It's like a spider web of food sources. But we can see 3 distinct circles down here at the bottom where my cursor is right now. And those 3 circles are the base of it all. In this case, they were epiphytes, phytoplankton, and macroalgae like sea lettuce. We can also see that there's this one node in the middle that seems to be connected to more other circles than the other ones. This species right here in the middle is called a keystone species. A keystone is in architecture the last stone in an archway that holds it all together. Take it out and the entire cathedral will crumble. It's like playing Jenga and you pull out the wrong brick, everything falls down. Now, the next step in this is to find out which actual species is the one that holds everything together. And I simply cannot give a presentation without showing at least a few pictures of worms. So here are some of the animals we regularly find around Cape Cod. This here is my absolutely favorite worm. It's called Prionospheus denstrupi. And to me, it looks like a dragon. And maybe you can see the similarities here on the face and the little cute eyes. Here we have another worm. Most invertebrate species don't have common names. So as my volunteers might know, we get to make up our own, internally only, of course. And this one we call mustache worm for obvious reasons. Then the next one we have is a scale worm. It's called Harmothoe. And if you're into Greek mythology, you might recognize that name as one of the Amazon warriors. And her shield had the same pattern as the scales of this worm, allegedly. Then this one is a fairly common worm. It's called Nereus or ragworm or clamworm. The Nereus refers to the Nereids, again from Greek mythology, the 50 sea nymphs, the daughters of Nereus. Here we have the cutest invertebrate ever, the tiny little horseshoe crab. And by little, I mean a few millimeters. Back here, you can see that the tail hasn't even developed yet. So this picture was taken just after hatching. And then I have one more picture of a sea urchin. It's only 2 millimeters long, but you can already see its mouth developed here in the center. and it looks just beautiful to me. So almost anything can act as a keystone species, from beavers to bees to prairie dogs. And they not only provide a food source for their predators, but they also can change the environment, and by building dams or pollinating plants or creating new habitats. And while a lot of terrestrial keystone species are well known and well loved, marine keystone species are often overlooked because they're small. But no matter how small they are, we will find them. Just like we did for the seagrass beds of around Cape Cod. Seagrasses like this eelgrass here are an important ecosystem. They provide cover and hiding spots for juvenile animals, which means that they usually boast of a high species diversity. There's a cell phone going off over there, sorry. In eelgrass beds around the Cape, the keystone species is an amphipod, an animal about the size of a grain of rice. Not a lot of people have heard of amphipods before. That's because they're not something we usually pay attention to. They're small, they're good at hiding in aquatic vegetation or in sediment, and they are of no direct commercial interest at all. Excuse me. But there's more than 9,900 species of amphipods. They live in salt water, they live in fresh water, they can even venture onto land. They're everywhere. Amphipods are food for larger animals and in turn keep algal growth and biofilm under control by their grazing. So this tiny little critter in the eelgrass beds of Cape Cod is unknowingly in control of its entire world. It's the make or break component of the eelgrass food chain or food web on the Cape. Without them, our food webs would crumble. But as I said before, anything can function as a keystone species depending on the ecosystem and the food web we're looking at. But what they usually have in common, at least in the ocean, is that they're small, inconspicuous, and not cute. To most people, to me, they still are. So from a conservation standpoint, if the make or break component of an entire ecosystem is only one species, protecting that one species means that the ecosystem should flourish. And here is the crux of the matter. Amphipods are too small to be protected individually. They aren't cute, they aren't majestic, there's no posters of amphipods in children's bedrooms. We might not be able to protect those small creatures, but we can protect their habitats, the eelgrass beds right on our doorstep, because every keystone needs an archway. So here I am fighting for something that I'm very passionate about, and I hope you will join me. Because every little habitat that's protected, every species that's protected, every piece of trash that's picked up can help protect the keystone species and might just save the ecosystem. Thank you.
That was terrific, Agnes. Thank you so much. I have a quick question before we get to everybody else's Q&As. I'm sorry, I just have to do this. How did you find yourself working with these tiny keystone species, these inconspicuous creatures, as you call them?
Well, I always liked the underdog. And I started to really look at them during my PhD in Germany when we tried to find out the most important components of seagrass beds. And it turns out that amphipods, they're everywhere, but they're easily overlooked because they're so small. But they are arguably one of the most important parts of seagrass food webs.
Well, and what about, you mentioned Benthic ecology, and how does what you're doing now tie into that?
So the benthos is the seafloor in general, comes from a Greek, again with the Greeks, comes from a Greek word. And the seagrass grows in the ground. There's other benthic habitats too. It's not just seagrass. Reefs can be considered benthic habitats. They are on the seafloor as well. Mangroves can be considered benthic as they grow out of the ground. There's hard bottom flats, which have a whole different kind of species assemblage, which also can be considered benthic habitat.
Fantastic. And are you doing some benthic habitat work off Provincetown right now?
Yes, we just finished sampling there, and we just started sampling in Marsh this past weekend.
That's great. That's a whole new project right there.
Yeah.
All right, so we'll move on to our Q&A from our viewers. First one from Phil. How do you spell the name of this keystone species? How often do they occur? For instance, are there hundreds of these per square meter in the eelgrass, or thousands, or something else?
So amphipod is spelled AMPHIPOD. It means pod, again from the Greek. Pod is means legs, and amphi means different kinds. They have different shaped legs, some for swimming and some for crawling. It's like amphi. Are there those cars that can go in the water and the land? So that's like amphipods, animals. And there's, depending on the season, they don't do well in the winter. The water gets too cold. But at the end of August, beginning of September, there can be thousands of them in eelgrass beds.
Wow, that's amazing. From Robert, what are the species one step out from the keystone in this ecosystem?
In the eelgrass, so that would be the species that eat the amphipods, and there's several small juvenile fish can eat amphipods. Sometimes isopods can get a hold of one. Isopods are the ones that look like pill bugs or wood lice. They usually crawl on the eelgrass blades. They can bite, so be careful. What else eats the amphipods? There's certain amphipods that live in the ground, in the sediment. Those can fall prey to worms, predatory worms. And then most amphipods are herbivores, so they graze off the biofilm on the eelgrass blades, or they get hold of decomposing algae that they can eat. Yeah.
Great. From Barbara, asking about keystone species in the salt marsh, can there be more than one keystone species?
Usually it's just one. There can be umbrella species. Umbrella is like an actual umbrella when it rains, which means that this one species is responsible or not responsible, can protect or can determine the fate of any other species that's below it, which doesn't necessarily mean it's also a keystone species. And there's engineering species like the eelgrass or oysters who actually form the habitat, ecosystem engineers, but that doesn't also necessarily mean that they are keystone species. But there's multiple important species in each ecosystem.
Okay, thanks, Agnes. We've talked about what eats the amphipods, but what do the amphipods eat?
They are plant eaters. They eat decomposing algae. They eat much like snails that graze on leaves. Yeah, small algae and biofilm mostly.
Okay. And back a little ways to the work that you're doing off Provincetown right now. We had a question about the eelgrass off the east end in Provincetown. It's looking as healthy right now as it has at different times of the year. Can you talk about that for us for a bit?
So the eelgrass is looking a little bit fuzzy right now. That's just the epiphytes that grow on the eelgrass. So epiphytes are plants that grow on other plants, like orchids grow on trees in the Amazon. That's completely normal, and as the season progresses, they increase, and the leaves that will come out next spring will be nice and squeaky clean.
Good. And another question from the audience: how important are the micro and myofauna in marine sediments as keystone species in the benthic ecosystem? And maybe you can explain what myofauna are as well.
Microfauna, anything that you need a microscope for, I would consider microfauna. Of course, there's different microscopes that are more powerful than others. But in the early 70s, Azam et al. Discovered what we now call the Microbial Loop, which is arguably the most important part of food webs in the ocean. It's still not fully studied. It's still not fully explained. And it's hard to do because it's mostly bacteria. So I can't really go out and collect a bunch of bacteria. My equipment doesn't work for that. But the microbial loop describes how anything that's decomposing in the ocean, is it an algae or is it phytoplankton and zooplankton that fall down? Is it a whale fall? Is it fish, squid, anything that's decomposing, brings back the matter, the organic matter to the sediment. And this organic matter that gets further decomposed by those bacteria, which make it in turn available to loop back into the food web. So nothing really is lost in the ocean, everything gets recycled. And without the microbial loop, we wouldn't have a productive ocean at all.
That might actually feed into our next question from David. How do we get biology into the biogeochemical cycling of carbon, nitrogen, phosphorus, and oxygen? That might be a question for another day, but.
Yes, well, briefly, since we're all built, and not just us, also the creatures in the ocean, everything has carbon in itself. As it gets decomposed, it's called POC, particular organic carbon, or particular organic matter, if we not just focus on the carbon. And the microbial loop helps bringing it back into the food web. If we're talking about blue carbon or greenhouse carbon, that's definitely something for a written answer that will probably be pages long.
Thanks, Agnes. What is the tool used to go from mass spec results and C15 tracing to that network that finds a keystone species?
So after analyzing the samples for stable isotope composition, it's basically a bunch of numbers. And those numbers get put into, right now I'm using a mixing model. It's based on Bayesian statistics, which works with probability. There's many models out there. Most of them are written in R, and most of them are free to anybody who knows how to use R. And the models tell me the probability for each individual animal that I'm looking at, what it ate at the point that I sampled it. And by that, I can determine the keystone species, because if one species shows up again and again and again for multiple animals that I'm testing, it's very highly likely that this species is a keystone species. And based on that, I can do more statistics and tests to see if this is true.
Great, thank you. From Christy, how do we go about saving these keystone species?
Well, for amphipods here on the Cape, we can definitely look out for eelgrass. They're very abundant in eelgrass. They're also abundant in mangroves. But the first step is to identify the keystone species. And then the second step is to identify the habitat this keystone species needs. Because it's not always an amphipod, it could be anything. And then we can focus on protecting that habitat.
Right. How much diversity is there among amphipods?
So there's worldwide, there's a total of 9,900 species, more or less. Here on the Cape, we found about 55 or so.
Yeah.
It's mostly worms here.
Yeah. And we're okay with that. Yeah. I did have one question, which is actually quite specific to our area here, because they're washing up on the beaches as we speak. How does salps fit into the marine ecosystem?
Salps are filter feeders. They float around. They can't really control where they swim. So salps are actually really interesting because you can trace where they were by analyzing what they ate. They usually eat microalgae, phytoplankton. So they're very low in the food web. They're very low in the food chain. And they don't, as they decompose again with the microbial loop, they sink to the bottom and then they basically get recycled and feed into the food web again.
Great. And one final question. Difference between seagrass and eelgrass, or is the one?
So eelgrass is one specific species. It's Softera marina. And seagrasses can be many, many species. We also have turtle grass in the Caribbean. It's very, very common. We have, I don't know the common names. There's another seagrass on the Cape that you might find in East Harbor. And you might also find it in Wellfleet Harbor. I can send around pictures of the other one.
That'd be great. Thanks, Agnes. I think that's it for questions from the panel. Did you want to add anything before we wrap up for today?
No. Just no. Everybody think about what their favorite invertebrate is.
Should we send in answers on a postcard?
Yes, please.
We shall do that. All right. Agnes, thank you so much. That was terrific. Everybody, thank you. Thanks for tuning in today. We will have this video ready to be posted online in the next couple of days. So if you do have any follow-up questions for Agnes, do email her at ccs@coastalstudies.org, and you can also find us on Facebook, Instagram, and Twitter. Oh, hold on, we do have one or two more things coming in. Abigail, horseshoe crabs.
Yes.
Not quite sure, but Abigail, if you want to expand on that, drop us an email and we will do what we can.
Horseshoe crabs are awesome.
Horseshoe crabs are awesome. Yeah. Oh, it's Abigail's favorite invertebrate.
Oh, thank you.
Excellent choice, Abigail. All right. Well, thanks so much, everybody. And we will see you next time on our Dockside Chat. Take care now. Bye-bye.